Hi, awesome nerdlings. I'm the Queen Nerdling, and today we're going to be discussing transcription and RNA processing. Let's go ahead and get started. So we're going to be talking about something that's called the central dogma. And this is really a two-step process with transcription and translation, so we can actually upregulate genes in the form of proteins. And these are all made from information that's stored in DNA. But that DNA has to have a way of being expressed. So before we really delve into transcription, I want to talk a little bit about the differences between RNA and DNA. So RNA is single-stranded and it has a lot of different types of structures, whereas DNA is double-stranded and it's a double helix. In RNA, we have nucleotides that have ribose sugar, whereas in DNA, we have deoxyribose sugar. RNA is less stable than DNA, and RNA is made in the nucleus, and it can actually leave in eukaryotic cells and go into the cytoplasm. DNA in eukaryotic cells stays in the nucleus, and it never leaves. We have many different types of RNA, and we only have one type of DNA. So we have three main types of RNA, although there are others. So first of all, we have messenger RNA. This is a single-stranded RNA. It's linear, and it carries codons. It's also the template for translation. We have transfer RNA, which is named after its T shape. It carries with it the anticodon as well as an amino acid. And it reads the messenger RNA codons and brings in those amino acids to the ribosomes to make proteins. We then have rRNA, or ribosomal RNA, and this is what composes the large and small subunits of a ribosome. So our messenger RNA over here, ribosomal RNA, and transfer RNA. And this transfer RNA, as you see, this little guy right here, is an amino acid. And so the transfer RNA carries the amino acid as well as the anticodon, and that anticodon will match up with the codon. So transcription is the process by which RNA polymerase, which is an enzyme, reads DNA and makes a messenger RNA copy. In eukaryotic cells, this occurs at the nucleus. So messenger RNA is an RNA copy of DNA. An RNA polymerase, and remember ACE means enzyme, reads the DNA sequence and it adds the complementary RNA nucleotide across from the DNA. So remember, we have certain base pairing rules. Adenine is going to bond with uracil, and cytosine is going to pair with guanine. We have our DNA sequence to our messenger RNA sequence. So in DNA, remember that we have adenine, thymine, guanine, and cytosine. Now in RNA, uracil is going to take the place of thymine. So when we're making that complementary messenger RNA, uracil is going to bond with adenine, not thymine. So as a short overview of transcription before we really delve into each of the little parts, transcription begins when RNA polymerase binds to a promoter upstream of the gene of interest that's going to be transcribed. In eukaryotes, we have the TATA box, which is just a sequence of thymines and adenines that serves as this recognition point for transcription factors, which are proteins, to bind. Now in eukaryotes, RNA polymerase is going to bind to the transcription factors and initiate transcription on that template strand. And so this process occurs in three different parts. We have initiation, elongation, and termination. So as far as initiation goes, we're going to have an RNA polymerase that binds to a promoter upstream of a gene. Now that promoter is a sequence that defines where transcription of a gene should actually start, basically saying, this is where we want you to start copying this gene or upregulating it. So we have this occurring upstream at the 5' end of the transcription site. of the 5' end of that 5' to 3' strand. So again, this is going to occur upstream at that 5' end of the transcription site. So looking at that right here. And then we're going to have that on that 5' end of this 5' to 3' strand. In both prokaryotes and eukaryotes, RNA polymerase is going to attach to the DNA by temporary hydrogen bonds with the sugar phosphate backbone of DNA. And remember that hydrogen bonds are easy to make and easy to break. So as far as initiation in prokaryotes goes, RNA polymerase can directly bind to the promoter region. Now this does not hold true for eukaryotes. In eukaryotes, we first have to have 5 to 6 proteins that are called transcription factors, which bind to the promoter. Then the RNA polymerase is going to bind to the proteins, and this eukaryotic promoter is called that TATA box, or the thymine adenine box. So next up is our elongation process. This is the process by which RNA polymerase lay the nucleotides. So we have our template DNA strand, which is running from our 3' to our 5' direction, and the non-DNA template strand, which runs from the 5' to the 3' direction. So elongation is pretty much the same in both eukaryotes and prokaryotes. The enzyme RNA polymerase breaks the bonds between the DNA base pairs, and it makes the messenger RNA strand as it moves along the DNA. And so as you see here, we have the RNA nucleotides being laid down by the RNA polymerase. Now this entire process is very different from DNA synthesis. In DNA synthesis, we have way more enzymes performing different functions, whereas in transcription, whenever we're synthesizing that messenger RNA strand, we have RNA polymerase, which is doing the roles of multiple different enzymes that we would have in the DNA synthesis process. So then we have our termination. So in RNA polymerase, it keeps adding these nucleotides until it reaches the end of a gene. And there are a lot of ways that transcription can actually stop. I came in like a wrecking ball, and I have no pitch at all. So that song might seem vaguely familiar, but just like a wrecking ball, a protein that's called rho-dependent literally comes in pretty much like a wrecking ball, and it knocks RNA polymerase off. So if you recall, RNA polymerase is attached to DNA by these temporary hydrogen bonds. Now those hydrogen bonds, again, easy to make, easy to break. They also have those bonds with that sugar phosphate backbone. Now the Rho protein, just like I so beautifully sang, basically smashes into RNA polymerase, breaks the bonds between it and the DNA, and it falls off and stops transcribing. So in both prokaryotes and eukaryotes, another way that this terminates is whenever we have a hairpin loop. So the bases at the end of a messenger RNA pair with each other. This forms a hairpin loop. RNA polymerase is prevented from actually moving forward. So after termination, what comes next? Well, it truly depends on if we're talking about prokaryotic cells or eukaryotic cells. Prokaryotic DNA doesn't really have a next. So in eukaryotic DNA, we have these different portions of the DNA called introns as well as exons. Now, introns stay inside of the nucleus. Exons are allowed to exit the nucleus. So while eukaryotic cells have both introns and exons, prokaryotic cells don't. So they're pretty much finished with the process. And in prokaryotic cells, transcription as well as translation actually occur simultaneously, whereas in eukaryotic cells, these two processes are separated. So in prokaryotes, since they lack introns, the messenger RNA is ready to be translated immediately. And like I said, prokaryotes couple transcription and translation at the same time. So this means that the messenger RNA is being translated by ribosomes into a protein while it's actually being made. Now, when it comes to eukaryotes, the process is a little bit more complicated, and we have an entire process of post-transcriptional processing of that messenger RNA strand. So we basically have this pre-messenger RNA strand, and we have to process it before it is able to exit the nucleus. And we do this with 3 different methods. We have to add a 5' cap or a G cap. We also have to add a poly A tail to the 3' end of the strand. And we have to splice out the exons. The exons are what gets to exit the nucleus and actually go to a ribosome to be upregulated, whereas the introns remain inside of the nucleus. So nuclear import and export is a very highly regulated process. This process prevents substances from getting in and destroying the DNA, but it also makes it very difficult for messenger RNA to get out unless it has special keys to unlock the nucleus. So the first key that's going to be added is polyadenylation, or a poly A tail. This is added to the 3' end of the strand, and it's made up of thousands of adenines. And this allows the messenger RNA to actually exit the nucleus. It also helps to prevent that messenger RNA from being destroyed by enzymes in the cytoplasm. Next, we have to add a 5' G cap. And so this is basically a modified guanine nucleotide that's going to be added over here to the 5' end. And just like that poly A tail, it's going to prevent the messenger RNA from being damaged by different enzymes in the cytoplasm, as well as act like a key to get out of the nucleus. Once we have that G cap and the poly A tail, we now have to do something about all of these introns and exons. So we know that the exons have to exit the cell, but we have to keep those introns inside of the cell. And we do this through a process called splicing, which occurs at a spliceosome. Once the spliceosome removes the exons, the introns remain inside of the cell. The exons are actually spliced together by a ligase, and then those are allowed to exit the nucleus. So this is our spliceosome right here, and it's going to remove the introns and splice together those exons. So it occurs in this complex, the spliceosome. The spliceosome is composed out of small nuclear ribonuclear proteins, or SNRNPs. These are enzymes that can form the spliceosome, which excises, meaning cuts out, the intron from the messenger RNA sequence. Now those introns stay inside the nucleus. The spliceosome is then going to splice together the exons using ligase, and then those are going to be able to exit the nucleus. And so it basically takes this intron right here, it's going to form a loop, and it's going to cut it out. These 2 exons right here, exon 1 and exon 2, are then going to be spliced together. Now remember that they also have those 2 keys that allow it to get out of the nucleus. They have that 5' G cap, as well as that poly A tail. Now we have the messenger RNA strand that's ready to exit the nucleus, carrying only the genes that are needed. So what's up with these introns and why do we have them? Well, scientists don't really have a concrete answer, but it does allow for a process called alternative splicing. So basically, multiple messenger RNAs can be made from each other. And this means that multiple protein variations can actually be made from the same gene. And that is the process of RNA processing in eukaryotic cells. So I hope that was helpful. For more videos like this one, you can go to www.nerdlingscience.com. This is the Queen Nerdling signing off for today.